So metagenomics is a really interesting field because when DNA sequencing prices came down, this whole field emerged. There's tons of bacteria on Earth, in our bodies, in every environment known, and we don't know anything about them because we could never really culture them in the lab. But with DNA sequencing being so cheap, what we can do is go into an environment, take a sample, and just isolate DNA, and we know that there are tons of bacteria that we can never culture. So, like I said before, DNA sequencing pricing is coming down, metagenomics is emerging, not only in the environment, but we're finding out things about human health that we never knew. So we know that when we're using antibiotics, in young children, that we change the microflora, and the human microbiome changes because antibiotics kill bacteria, and what we're seeing is this can have an influence on human health. So we have metagenomics in human health, we have metagenomics in the environment. But what's also interesting is metagenomics is fueling a whole new era in synthetic biology. So with sequencing prices going up, what we're seeing is we're now starting to synthesize pieces of DNA, which are probably limited to about 500 base pairs to longer and longer base pairs. So what we'll see is synthetic biology really go through the roof in terms of applications, and it's being fueled by the gene discovery in metagenomics. Well, first of all, I think this event was very good. It was a 2-day event, and it was in metagenomics. Not so much research areas, but in application and in bioinformatics training. Because I think what's interesting about metagenomics is it's such an emerging field because DNA sequencing pricing has come down, that we're generating tons of data, and we actually have no real idea of how to analyze it. We're just developing these methods. So this meeting comes at a real timely a timely time, I guess, because we brought people together that were really experts in the field and sort of look at methods that they're developing to do data analysis. So that was sort of one day, day one. And on day two, we have training going on with hands-on. So we have 100 people in a computer room actually working with these experts and who develop these tools. So it was very well attended and very exciting to actually do this in Waterloo. The major opportunities for metagenomics, well, first of all, we have tons of sequencing facilities, and we have a great history of environmental research in Ontario. So I think bringing these technologies that are emerging from the health field to sort of the work on problems in the environmental field are great. And I think Ontario is poised because they have a lot of sequencing capacity, and they have a lot of really cool environmental problems that they're trying to solve. So merging these two fields and networking this community here with big consortia outside of Canada that are from a bunch of different countries, getting them all together to talk and create standards in a field that's really rapidly developing is sort of a great opportunity for Ontario researchers to really be a leader in environmental metagenomics.